going to present me is just, my name is Raquel. I'm a PhD student in biology, in molecular and cellular research. But today, the work that I'm going to present you is about a collaboration that I did with the Max Planck Institute for Dynamic Sense of Organization. The PI of this project is Dr. Vahid, but he was not able to be here today, so I'm here just to transmit a little bit of information. And today I'm going to talk to you about the movement of cells in confinement and also in low adhesion substrates. Just a general overview about cell migration. Cell migration is only the movement of cells, is a very important mechanism approach in vivo in humans, since the embryonic development to tissue regeneration, for wound healing, also for the immune response. In the literature, there are two main types of cell migration that are described, that are the mesenchymal migration and the amoeboid migration. These types of migration depends on the physical environment that the cells have. Usually when they have substrates with adhesion, they have this type of migration, which they are able to do adhesions with integrins, focal adhesions, and other proteins of the extracellular matrix. Then the cell is able to contract and move. However, in the cases where we don't have a good adhesion, the cells depend on the contraction of the cortex, depends also for intracellular pressure to stabilize a bleb that allows the cells to move. However, there's a problem with these approaches, is that usually these types of studies are done in two-dimensional structures, usually in cell culture. And the problem is that in vivo we have vessels, we have interstitial pressure. So usually in vivo the cells are in confinement and low adhesion environments. So there's some studies in the literature trying to simulate this behavior and this model of low confinement and low adhesion. And specifically in this study, they try to understand how the contractility of the cortex generates this amoeboid migration. And the main results that they obtained here was that the confinement, when the cells are in confinement, amplifies the cortical contractibility gradient, and this drives the polarity in the cell and then to blebbing. Very interestingly, in the same year, in the same journal, in the same issue, another work was published saying that this type of migration depends on the switch of the phenotype of the cells from mesenchymal to amoeboid. So they conclude here that the cells under confinement are able to switch their mechanisms and switched from mesenchymal to an amoeboid migration. There's another studies on the literature showing the importance of proteins and mechanisms under these two types of migration, but this is our point. This is more biological, and in this project we are focused on the physics. So in spite of the literature, the full characterization of the relation between the confinement and motility is not known. So there are several questions that we needed to understand how the cells move in this type of behaviors. So to answer to this question, this is a little bit spooling, but basically here is the model that we use it for our experiments. Basically, we put one drop of cells between two functionalized glasses with a polymer, and then we flatten the cells, and with a distance of 1 micrometer, that is a very confinement limit, and then we did some imaging. So after we flatten the cells, the first question that we ask is actually how do cells react when exposed to this confinement. So for that we did time-lapse imaging, and in this first image, we can see that immediately after flattening, appears small protrusions around the cell surface of the cell. These type of structures are called blebs. If we zoom in into one of these blebs, and interestingly we found this type of analysis in all the cases that we studied, is that these blebs appear in a region of cortical detachment. So after the bleb appears, the actin is recruited for this region, and after a period of time, the cells start to contract, as we can see just representative in this graph. Indeed, is only to show you that the time where actin is recruited is very shorter compared with the remaining time that the cell is contracting. We found another scenarios in the cell after we flattened, and in this second scenario, we can see here this is a bleb, this is the remaining cell body of the cell, and we can see that this little bleb is expanding from the cell, but at the same time, this part of the cell is contracting to maintain the overall stability. Then this bleb is just continuously growing, forming a more larger and organized structure. After a short, there is an increase of active recruitment between this connection between the little bleb and the remaining body, that we called this structure the constrictive neck. However, we saw that cells that are not able to form this little structure and the constrictive neck, instead of forming these processes, one single bleb forms multiple larger blebs that just expand and expand over the time until the cell ruptures. So actually, by these results, our next question was, okay, is this actin neck only having an impact in changing the cell shape of the cell, or is actually helping the cell through another mechanisms to survive? For that, we did some quantification, and in fact, we saw that cells that are able to form this structure and this constrictive neck are able to maintain their normalized area during more time. However, the cells that don't form this ring just expand over time and rupture very shorter. Here is basically the representation of this that I told. The time higher from cells that are able to form this information, in contrast with a time that these cells without the little bleb just get not viable and die. So in fact, what we concluded from this data is that the actin neck acts a mechanical stabilizer of the cell, but also is delaying the rupture by preserving the cell integrity. Then we wanted to understand how actin reorganizes during this process of blebbing, and at the beginning we can see here in image A that the actin is diffuse, broadly distributed. However, this is more clear in the video. Here I only have the photos, but it's very clear that more or less after 30 seconds starts having an accumulation of actin in the base of the bleb. And this pattern of intensity of actin is very clearly showed here by the intensity profile of actin, which peaks with more defined after 30 seconds. This is just the schematic representation of the blebbing that I told you. So at the beginning we have the bleb, and then the actin starts concentrated into the space of the ring, and the cell starts to retract. So in this image, in these images, I show you a little bit about the actin flow that we saw inside the cells. And here we can see that once again the little bleb, the remain body, the accumulation of actin in this constricted neck, and it's possible to see an actin flow from this part of the cells towards the constricted neck. It's like a flow like that. We did chemograph analysis just to quantify and to describe this type of flow, and we found velocities of this actin flow around 0.24 micrometer per second. The next images are from polarized microscopy, where we see and where we can see that the constrictive neck is here accumulating the actin in this point to limit the movement of the cytoplasmatic material from the cell body into the blood. After a short period of time that the actin is accumulated here, the cell starts to contract towards the cell body. So in fact, it is possible to see that this actin flow is not only contributing to increase the constriction of the neck, but also is pulling the blood toward the cell body to protect the cells. However, after a period of time, the ATP lasts in the cell, so the actin is not more able to recruit into these regions, and the cell just start to expanding until rupture. So until now, what you understand for that and what I'm trying to explain to you is that all these mechanisms that we see, all these effects that we see, have one main intermediator, that is actin. It's actin that is promoting all these processes. So to take care of that, we did one thing that was inhibited actin from the cells. And when we treated the cells with latrampolin, that is a drug that inhibits the polymerization of actin, we saw that the cells lose the capacity to form this little structure and just adopt a softly and globe morphology. As we can see in this graph, and this is only the representation of the increasing expansion and a shorter periods of times until that. We're trying to understand if partial inhibition of actin also had these results. We saw that at the beginning the cells are able to form these blebs, but after a short period of time the cells just have a similar morphology as when we treated with high concentrations of latrampolin. So the main conclusions from these images is that, as we know, the cortex is very dynamic. So we conclude that when the cortex is damaged, a bleb appears in this region. Then, of course, actin recruitment for this region, and the cells that are able to form this structure are able to survive for a longer period of time. Our next question, final question, was to understand if exactly in this model we can have the movement of the cells, we can see self-propulsion of the cells. Here once again is our leading bleb, the remaining cell body, the accumulation of actin into this point. Here we can see several irregularities in the bleb that shows the active dynamic and the active flow inside. And then we actually found that the cell is moving in opposite directions that the actin flow. So basically, in this model, under confinement and in non-adhesion, the neck contraction generates internal forces that pull the cells in opposite direction of the actin flow and towards the cell body. Just to explain you, in very simple words, when we swim, it's like we do like that. It's like that is exactly the same with actin flow for that, but our body is to opposite directions, and this is basically what we found. Then Vai just tried to do some simulation to to characterize a model for these processes. And basically what this diagram means is that the cell is converting the internal tension into motion. Basically, here in the region of the neck, where the active flow is in this direction, so here in the neck we have a higher tension, so we will need to apply less pressure to do the suction moves. Here just the schematic representation, once again showing the accumulation of actin here, the flow of actin and the movement of the cells in opposite directions. So just to conclude, I put here one video where it's possible to see the real effects when we see the formation of the blebs shortly, and then this active flow here and the contraction to protect the cell of rupture. Basically, by this work, we answered to these main questions that were missing in the literature, that basically why does a little bleb emerge, when it becomes dominant, and how these main effects contribute to the movement of the cells. So our message, the main message that we wanted to give you by this work is that in fact, when we have this type of eye confinement and no adhesion, is not a capacity of the cell of switching the phenotype, but this is only a physical phenomenon. It's only the actin flow that promotes these forces inside the cells that makes the cell propelling in opposite directions. So, yeah, just to finish, just to thank you, all the team. This is the PI of the project. I put also his email here, because if you have some question, of course, he seems the best person to answer your questions, and maybe also if you have some ideas, something to add, you can also contact him. Thank you very much.
For questions.
I'm not a biologist. Is that a general phenomenon? When we put pressure on the cells, they develop this neck and leg and everything. It happens in all cells.
Yeah, we tested this in different cells because it's described that usually our, for example, cancer cells are able to switch the phenotype to survive in tumor microenvironment when we have a high concentration of vessels and so on. And for that, we did in 3 different prostate cancer cell lines, and we did also in different epithelial cells and also fibroblasts, and we found the same response in all the cases. So yeah, it's in all the cells.
I was wondering if you can make an analogy with glaucoma. In glaucoma, there is pressure on the retina. There is pressure on the retina and there are problems of retina detachments and those the other.
Yeah.
Do they reported any sort of similar behaviors, like these effects which come due to the pressure?
Yeah,
confinement, pressure.
Exactly. It's this confinement, this pressure that allows cause damage in the cellular cortex, and is because of this damage that then the bleb appears, and then the actin flows to maintain the stability of the cell. So yeah, the main cause of this phenomenon is pressure. It's the pressure that then leads to cortical actin damage.
Is this published anywhere?
We submitted this work last week to Cell. We are waiting now for the One of the questions I think that the revisers could ask us is about, because we did also some, I don't put here because it's very difficult in the final of our experiments, because we put 4 microliters of cells between 2 glasses, and then it's very difficult to collect the cells to do the other analysis. So we don't have enough data to show you, but we have preliminary data showing that the cells before and after flattening don't have changes in these proteins involved in mesenchymal and amoeboid migration. So,
And if you remove the pressure, would the bleb retract?
if we remove the pressure,
Yeah.
The study that we did is that after we flatten the cells and we do all the analysis, we separate the classes, we collected the cells, and we evaluated the viability of the cells. And this viability is conserved even after extreme flattening. So about your question, if we remove the pressure, the bleb retracts. Actually, we don't need this type of experiment, but I think that if the cortex remains damaged, the cell will try to have this mechanism to recover.
In the interest of time,
Short question. Are these blebs related to inhomogeneities in the membrane? I mean, they appear where the membrane is more thin or
Damaged.
something or
Yeah.
damaged.
Yeah, this was one of the things that we noted is that these blebs only appear in regions where there's the damage of the cortex. Otherwise, they don't develop in this way.
How many intermediate filaments involved in this bleb formation?
Actin filaments.
No, intermediate.
Yeah.
The filaments of 8 to 10 nanometers.
Yeah. About to answer to this question, we used also an inhibitor of these intermediate filaments, but we didn't see these alterations that we saw when we treated the cell with lactambutin. So when we target the actin, the cells lose these effects. When we target the intermediate filaments, we don't see the same effects.
That's
So
not
it seems,
important.
yeah, it seems that it's actually the actin that is important in this process.
so 2 more questions. Sure. Yes, I was just curious whether you did notice morphological deformation of the nucleus as well.
This is work that we are trying to do, but we are facing some problems because it's very difficult to see alterations of the actin inside the nucleus. So yeah, we are working on that. Now we have another models of cells with some mutations and so on. And yes, but for this analysis, we want also to analyze the differences in nucleus, but until now we don't have enough results to show you how this dynamics occurs in the nucleus.
Yeah, thanks. So you see in your assay that has no adhesion, if I understand correctly, that the center of mass transfers, right?
Yeah, exactly.
So you calculate the total center of mass and it does propel.
Yeah, we use it specifically the glasses functional as a metapolymer.
Yeah.
It's just to ensure that we have a similar approach in all the glass and that we don't have adhesion.
My question actually is it then a gliding assay? Because I mean, you need to transfer some momentum to the environment to propel the cell, right? If you have no adhesion, how does the cell move? Does it have a gliding surface assay? Is it gliding mechanism?
It's exactly because in the mechanism that in amoeboid migration, the cells don't need to use these adhesions to move. It's just the actin flow that is propelling the cell.
But you do need an outside flow, otherwise you don't get any forward.
And outside, we
Something
have.
has to move on the outside, otherwise the inside will not transmit.
Yeah, we have. When we put the cells under the 2 glasses, we see the cell movement, but it's not during for this flow. It's from the flow from the environment. But after a period of time, we optimize our technique to stop the flow outside the cell. So you put the cell, and the liquid, after a while, just you stop the flow.
Okay, thank you.
Just because I'm the chair, no? Can you go back to the last video you showed? And could you please comment a little bit on where do you think occur the extra actin that comes from the outskirts of the cell?
From the outside spots.
Once now you see you start here.
Yeah, for example.
Yeah, okay, but now you'll see the larger ones that are.
Yeah, here in this video, I have another videos with a little bleb, like the previous examples that we saw that after a period of time there is a bleb that is dominant. Here I put this video just to show you that, because at the beginning we see different blebs, and I will show you when they're acting. I can stop maybe. But it's in these points. You see here, you see the development of the blood, the recruitment of actin into there, and then occurs a flow of actin in this direction, and then you see the constriction there.
Yeah, but you have also a very large skirt that comes now here. You see, where does it come from?
Yeah, this is the. Are you meaning this structure?
Yeah.
This is when the cell lost the ATP and is not more able to recruit actin into these regions, and after this the cell achieves this length, the cell died.
But could it be possible that, for instance, if you have the two slides that are equally functionalized, then you have like a clepsydra stuff, and what you're actually seeing in the fluorescent microscopy is how the outer surface of the cell that was already attached on the top slide can just enter into your visual level.
No, when we are taking these videos, we have different bubbles that could appear in the medium, but we are sure that it is actually, this approach is very difficult because you need, for example, sometimes you start a visualization of the cell, and after some hours a bleb appears, so you can't do quantification and analysis. So this was a thing at the optimization of the process at the beginning to take sure that these things not happens in our system.
Thank you very much.
Okay, thank you.